Welcome to JRF Academy. In this video we work through 5 original CSIR NET pattern questions from Unit 2. Cell Biology. These questions are designed to match the reasoning demands of Section B and Section C of the CSIR NET Life Sciences examination. Our 5 topics are the retinoblastoma protein and E2F regulation at the G1S checkpoint, receptor-mediated endocytosis and inhibitor pharmacology, differential centrifugation for lysosome enrichment, the 3 major cytoskeletal systems compared side by side, and the crosstalk between the extrinsic and intrinsic apoptotic pathways. Each answer is explained with stepwise reasoning, the core concept, a concept diagram, and a full distractor analysis using the JRF blueprint. 3 questions carry 4 marks each. Section C pattern, and 2 carry 2 marks each. Section B pattern. Let us begin. Question 1. Section C pattern. 4 marks. Assertive type. The retinoblastoma protein, ROB, is a tumor suppressor that guards the G1S boundary of the cell cycle. Consider the following 4 statements about ROB function. Statement A. In quiescent cells, hyperphosphorylated ROB binds E2F and recruits histone deacetylase, silencing S-phase genes. Statement B. Sequential phosphorylation by CDK4/Cyclin D, then CDK2/Cyclin E, creates a positive feedback loop that drives irreversible S-phase entry. Statement C. Loss of ROB function causes permanent cell cycle arrest because E2F is constitutively repressed. Statement D. At mitotic exit, protein phosphatase 1 dephosphorylates ROB, restoring its ability to bind E2F in daughter cells. Which combination of these statements is correct? Option A, B, and C only. Option B, C, and D only. Option C, A, B, and D only. Option D, A, C, and D only. Pause and think before proceeding. The correct answer is option C. Statements A, B, and D. The core concept. ROB is the molecular brake that prevents premature S-phase entry. In quiescent cells, ROB is hyperphosphorylated and bound to E2F. This complex recruits histone deacetylase to silence genes encoding cyclin E, pCNA, and other S-phase components. Growth factor signaling activates CDK4/Cyclin D, which phosphorylates ROB at select serine and threonine residues, partially releasing E2F. Partial E2F release induces cyclin E transcription. CDK2/Cyclin E then further phosphorylates ROB in a positive feedback loop, creating the bistable switch that makes restriction point crossing irreversible. This is precisely what statement B describes. At mitotic exit, cyclin B is degraded by APC, C, CDK1 is inactivated, and protein phosphatase 1 dephosphorylates ROB, resetting it in daughter cells. Statement D is correct. Now, statement C. ROB loss causes arrest. This is completely wrong, and it is a classic high-stakes distractor in CSIR NET. ROB loss means E2F is constitutively active, not repressed. Without the brake, cells proliferate. Without restraint. This is the mechanism of retinoblastoma, a childhood eye cancer caused by biallelic ROB deletion. Statement A. TRUE. Statement B. TRUE. Statement C. FALSE. Statement D. True. Options A and D include the false statement C. Option B drops the true statement A. Only option C correctly pairs A, B, and D. The answer is C. Question 2. Section C pattern. 4 marks. Analytical type. A cell biologist measures the uptake of 2 cargoes. Transferrin, which is taken up by Clathrin-mediated endocytosis, and cholera-toxin B subunit, which enters via caveolae, and lipid raft-mediated endocytosis. After separately treating cells with 4 inhibitors, filipin sequesters cholesterol in the membrane. Dynasore inhibits the dynamin GTPase. Pitstop 2 blocks the Clathrin terminal domain. Methyl-β-cyclodextrin acutely depletes membrane cholesterol. Which one of the following correctly predicts all 4 experimental outcomes? Option A. Filipin blocks cholera-toxin B only. Dynasore blocks both. Pitstop 2 blocks transferrin only. Methyl-β-cyclodextrin blocks cholera-toxin B only. Option B. Filipin blocks both. Dynasore blocks cholera-toxin B only. Pitstop 2 blocks both. Methyl-β-cyclodextrin blocks transferrin only. Option C. Filipin blocks transferrin only. Dynasore blocks both. Pitstop 2 blocks cholera-toxin B only. Methyl-β-cyclodextrin blocks both. Option D. Filipin blocks both. Dynasore blocks transferrin only. Pitstop 2 blocks cholera-toxin B only. Methyl-β-cyclodextrin blocks both. The correct answer is option A. The core concept. Clathrin-mediated endocytosis uses Clathrin coat proteins, the AP2 adapter, and dynamin for membrane scission. Caveolae-mediated endocytosis uses cholesterol-rich lipid rafts, caveolin1, and also requires dynamin for membrane scission. The critical insight is that dynamin is a shared pinchas required for both pathways. Cholesterol disruption specifically collapses caveolae but leaves Clathrin pits intact. And pitstop 2 is Clathrin specific. Caveolae are entirely Clathrin independent. Step by step. Filipin is a polyene antibiotic that sequesters cholesterol by forming insoluble complexes. This disrupts lipid raft organization. Caveolae collapse and cholera-toxin B uptake is blocked. Transferrin takes the Clathrin-coated pit route, which is cholesterol independent. Unchanged. Dynasore inhibits dynamin GTPase activity. Without dynamin, neither Clathrin-coated pits nor caveolae can undergo membrane scission and pinch off. Both transferrin and cholera-toxin B uptake are blocked. Pitstop 2 blocks the Clathrin terminal domain. Clathrin cannot form a coat. Transferrin uptake is blocked. Caveolae have no Clathrin. Cholera-toxin B uptake is completely unaffected. Methyl-β-cyclodextrin extracts cholesterol from the outer leaflet. Same functional consequence as filipin. Caveolae disrupted. Clathrin pits intact. Cholera-toxin B blocked. Transferrin unchanged. Option A correctly assigns all 4 outcomes. Options B, C, and D all reverse the dynamin or Clathrin assignments incorrectly. The answer is A. Question 3. Section B pattern. 2 marks. Quantitative type. A liver cell homogenate is subjected to sequential differential centrifugation. Spin 1. 600 times gravity for 5 minutes. Producing pellet P1. Spin 2. 3000 times gravity for 10 minutes. Producing pellet P2. Spin 3. 10,000 times gravity for 20 minutes. Producing pellet P3. Spin 4. 100,000 times gravity for 60 minutes. Producing pellet P4. A researcher measures acid phosphatase activity, a lysosomal marker, across all 4 pellets. In which pellet is acid phosphatase activity highest? Option A. P1. The 600 times gravity pellet. Option B. P2. The 3000 times gravity pellet. Option C. P3. The 10,000 times gravity pellet. Option D. P4. The 100,000 times gravity pellet. The correct answer is option C. P3. The core concept. Differential centrifugation separates organelles by size and density. Larger, denser structures sediment at lower centrifugal force. Smaller, lighter structures require progressively higher speed. Nuclei are the largest organelles, about 5 to 10 micrometers, and pellet at just 600 times gravity. They form pellet P1, along with unbroken cells and large cell debris. Mitochondria are smaller, roughly 0.5 to 2 micrometers, and pellet at 3000 times gravity. They are the dominant component of pellet P2. Lysosomes are smaller still, about 0.1 to 0.5 micrometers. They require 10,000 times gravity for 20 minutes to sediment. P3 is therefore enriched in lysosomes and peroxisomes. This is confirmed biochemically. Acid phosphatase activity peaks in P3. Acid phosphatase is a lysosomal enzyme that cleaves phosphate groups from substrates at acidic pH. A classical lysosomal marker used since the work of Christian de Duve. At 100,000 times gravity, the P4 pellet contains microsomes, fragments of endoplasmic reticulum, and ribosomes. By this point, lysosomes have long since been removed in P3. Option A. P1 contains nuclei. No lysosome enrichment. Option B. P2 contains mitochondria. Option D. P4 contains microsomes and ribosomes. Lysosomes are absent by this step. The answer is C. Question 4. Section C pattern. 4 marks. Comparative type. 3 major cytoskeletal systems underpin eukaryotic cell architecture. Match each element in column I with its correct property set in column 2. Column I. Actin microfilaments. 7 nanometers. E. Microtubules. 25 nanometers. E. Intermediate filaments. 10 nanometers. Column 2. Property O. Dynamic instability. GTP hydrolysis. Nucleated by gamma tubulin ring complex at MTOC. Property B. Nonpolar. Coiled coil dimer subunits. No NTPase activity. Highest mechanical rigidity. Property C. Polar barbed and pointed ends. ATP hydrolysis. Treadmilling. Branching by ARP 2/3 complex. Option A. I with C. II with A. IE with B. Option B. I with A. II with C. IE with B. Option C. I with C. II with B. IE with A. Option D. I with B. II with A. IE with C. The correct answer is option A. The core concept. Each cytoskeletal system has a unique biochemical identity. Actin microfilaments are polar. A fast-growing barbed plus end and a slow-growing pointed minus end. G-actin monomers add using ATP, which is hydrolyzed to ADP after incorporation. The ADP actin at the pointed end depolymerizes, creating treadmilling net flux from pointed to barbed end. The ARP 2/3 complex, activated by WASP or WAVE family proteins, nucleates branched actin networks at 70 degrees, building the lamellipodial meshwork that drives cell migration. Microtubules are built from alpha-beta tubulin heterodimers. Beta-tubulin binds GTP, which is hydrolyzed after incorporation. When the GTP tubulin cap is maintained, the microtubule grows. When the cap is lost, the GDP tubulin lattice collapses. Catastrophe. Readdition of GTP tubulin restores growth. Rescue. This is dynamic instability. Nucleation occurs at the minus end, anchored at the MTOC by the gamma-tubulin ring complex. Intermediate filaments are assembled from coiled coil dimers. Vimentin. Keratin. Nuclear lamins. They form antiparallel tetramers with no polarity, no nucleotide triphosphatase activity, and no treadmilling. They provide exceptional tensile strength, the most mechanically stable of the three systems. Option B reverses actin and microtubule assignments. Option C assigns coiled coil properties to microtubules. Option D assigns intermediate filament properties to actin. Only option A correctly assigns actin to C, microtubules to A, and intermediate filaments to B. The answer is A. Question 5. Section B pattern. 2 marks. Comprehensive type. This question tests your integrated understanding of 2 apoptotic pathways. Which one of the following statements correctly describes the relationship between the extrinsic death receptor pathway and the intrinsic mitochondrial pathway of apoptosis? Option A. In type 1 cells, active caspase 8 directly cleaves proliferation without requiring cytochrome C release, and BCL2 overexpression cannot rescue these cells from extrinsic apoptosis. Option B. BCL2 overexpression blocks both extrinsic and intrinsic apoptosis equally in all cell types. Option C. Caspase 9 is the initiator caspase of the extrinsic pathway, activated at the death-inducing signaling complex by Fas-Fas ligand interaction. Option D. The intrinsic pathway is initiated by Fas-Fas ligand binding, which triggers mitochondrial outer membrane permeabilization. The correct answer is option A. The core concept. Cells executing apoptosis via death receptors fall into 2 functional types. In type 1 cells, which include thymocytes and Jurkat T cells, the death-inducing signaling complex assembles efficiently and generates abundant active caspase 8. This caspase 8 directly cleaves PROCASPASE3 without any mitochondrial involvement, because BCL2 acts on the mitochondria, preventing Bax-mediated pore formation and cytochrome C release. It has nothing to block in type 1 cells. BCL2 overexpression therefore cannot rescue type 1 cells. This is a critical and frequently tested distinction in CSIR NET cell biology. In type 2 cells, including hepatocytes, the disc generates insufficient caspase 8 to directly activate caspase 3. Instead, caspase 8 cleaves BID to generate truncated BID, called TBID. TBID activates Bax and BAC, causing mitochondrial outer membrane permeabilization. Cytochrome C is released, forming the apoptosis with a PAF1 and DATP, activating PROCASPASE9. Then caspase 3. In type 2 cells, BCL2 can rescue by blocking permeabilization. Option B is wrong. BCL2 cannot rescue type 1 cells, so it does not block extrinsic apoptosis equally in all types. Option C is wrong. Caspase 9 is the initiator of the intrinsic pathway, not the extrinsic pathway. Caspase 8 initiates the extrinsic pathway. Option D is wrong. Fas-Fas ligand initiates the extrinsic pathway. The intrinsic pathway is triggered by internal stress signals. The answer is A. Excellent. Let us revisit the 5 reasoning frameworks from this cell biology session. Question 1. RORB and E2F. RORB loss releases E2F, driving uncontrolled proliferation. RORB loss does not cause arrest. This is the retinoblastoma mechanism. The positive feedback loop from CDK4/Cyclin D through CDK2/Cyclin E makes restriction point crossing irreversible. PP1 dephosphorylates RORB at mitosis exit. Question 2. Endocytosis. Dynamin is the shared GTPase required for both Clathrin-mediated and caveolae-mediated endocytosis. Cholesterol disruption collapses caveolae only. Pitstop 2 is Clathrin-specific and does not affect caveolae. Question 3. Differential centrifugation. Organelles pellet in order of decreasing size. Nuclei at 600 grams. Mitochondria at 3000 grams. Lysosomes at 10,000 grams. Pellet P3. And microsomes at 100,000 grams. Acid phosphatase peaks in P3. Question 4. Cytoskeleton. Actin uses ATP and treadmills. Nucleated by ARP 2/3. Microtubules use GTP and show dynamic instability. Nucleated by gamma Turk. Intermediate filaments use no NTP and show no dynamics. They provide mechanical rigidity. Question 5. Apoptosis. Type 1 cells bypass the mitochondria. BCL2 cannot rescue them. Type 2 cells require mitochondrial amplification via TBID. BCL2 blocks this step. Caspase 8 is always the extrinsic initiator. Caspase 9 is always the intrinsic initiator. For more CSIR NET pattern questions, practice at gekjrfacademy.com. Subscribe for videos covering all life sciences units. Thank you for watching, and best of luck in your examination.